Hello, welcome back. We are continuing our journey in chapter 10. We're going to get started with this mini lesson on inhibition of cell wall synthesis. So if you remember, we kind of ended on the last video with our goals for most of the rest of this chapter. What are the targets of antimicrobial action? So this mini lesson is going to focus on these drugs here that can target the cell wall synthesis. So if a cell cannot make its cell wall, it is susceptible to osmotic pressures and changes. It's lost its structural integrity. It basically is going to fall apart. There's a listing here: penicillin, cephalosporins, vancomycin, bacitracin, isoniazid, and ethambutol. Again, like I said at the end of the last video, I'm not going to be requiring you to know which drug does which particular thing. More focus on what is it that is being done to the cell wall that's going to prevent it from being able to be formed properly. So in the notes it talks, or in the book, in the notes in the book talk about different parts of the peptidoglycan cell wall that are targets. So for example, if you remember from chapter 3, I think we talked about the cell wall, and peptidoglycan is the large the protein carbohydrate combination, this big thick what's the word I'm looking for? Polymer that bacteria make their cell walls out of. So if you remember, it has these alternating units of NAM and NAG. So the NAM, NAG, that's the brown and the pink, And then we also have those little blue kind of tetrapeptide cross bridges, and then those amino acid linkers. So those are targets. The reason why peptidoglycan is such a good target for antibiotics is because we don't have it. So our eukaryotic cells do not have peptidoglycan. So something that's going to inhibit the production or synthesis of peptidoglycan will not affect our eukaryotic cells. Another thing about the inhibition of cell wall synthesis for drugs to be functional or effective is they really only work on growing bacteria. So if you're a bacteria and you already have your peptidoglycan cell wall made, any of these antibiotics are not going to make you die. Your cell wall is made. They don't penetrate in and break it up that way. The way that these drugs work is they prevent the coming together of the components. So some of them work by preventing the cell from actually making the NAM and NAG subunits, or being able to hook them together, or blocking the tetrapeptide, the amino acid cross bridges. So a cell would have to be actively growing and making its cell wall, these drugs to be incorporated into it, which is going to ruin the integrity of that cell wall. So we're going to focus primarily on the penicillin model of inhibition of cell wall synthesis. So this is done, penicillin has a component in its molecular structure. So here's penicillin right here, natural penicillin that Alexander Fleming found from the fungus. And in that molecular structure, it's got this little square-shaped part of the molecule called the beta-lactam ring. This word right here, that green square. So that beta-lactam ring is what makes penicillins effective in killing bacteria. And you'll see there's others. There's carbapenems, cephalosporins, and then the semisynthetic methicillin. And then there's a whole family of semisynthetics and synthetic penicillins. But if you'll notice, they all have the beta-lactam ring. That's the functional part of these molecules. So when a bacterial cell is growing in the presence of any of these drugs with a beta-lactam ring, it kind of inserts itself where those peptide cross bridges would be. So you can kind of see the little blue connections there. That's where they would supposed to be. But then you get these little molecules with the beta-lactam rings kind of inserting themselves, so the NAM and NAG subunits do not get hooked together properly. And so then we can see over here in this picture, it's not a strong cell wall. I like to use the analogy of, like, if you're building a brick wall, you have each of your bricks and then you mortar them together with the mortar, and then you can grout, and it's a nice solid structure. Well, what if you were to build a brick wall without any mortar? You're just stacking the bricks. It's not going to be a very strong cell wall. You can just, a brick wall, you can just push it over. So that's what happens with bacteria growing in the presence, bacteria with peptidoglycan, growing in the presence of these beta-lactam drugs, is their cell walls cannot be made properly. And so they are susceptible to osmotic. So here's a micrograph of an intact bacterium on the left, and then one that has been grown in the presence of penicillin or something with a beta-lactam ring on the right, that it was susceptible to osmotic changes. So the cell wall isn't very strong. Even the normal changes in hypertonicity or hypotonicity, which would be able to be withstood by a normal cell wall, would not be able to in one of these weakened cell walls because of the presence of the beta-lactam drugs. All right, so that wraps up our discussion on inhibition of cell wall synthesis and the first target of our 6 that we're going to be talking about of how antimicrobial drugs affect microbial growth. Okay, see you next time.